Good afternoon, everyone, and thank you very much for the invitation to the Cure Parkinson's Trust. I'm very honored to be here and be given the task of presenting an overview of the field to you regarding stem cell therapies for Parkinson's disease. As Helen so kindly introduced, my name is Agnete Kirkeby. I've been at Lund University for many years, working with Anders Björklund and Malin Palmer and many other people at the team developing cell therapy for Parkinson's disease, which has entered clinical trial in the Sweden and the UK. I'll tell you a bit about that journey, as well as give you an overview of other stem cell trials that are ongoing. We have in the room also other esteemed guests like the Kyoto team that also have a clinical trial they've just completed and published on, and I'll give you a brief overview of that also. Okay, so put very simply, what I'm talking about to you today is putting new neurons into the brains of individuals with Parkinson's disease. And what we're looking at here is replacing the dopamine neurons that are lost during the course of the disease. This image is actually a bit misleading because the neurons are lost in the substantia nigra, as many of you probably know already. There is this loss over time of the dopamine neurons located in the substantia nigra, but when we transplant new neurons, we actually put them into the putamen and not substantia nigra. The putamen is the site in the brain where the dopamine is released, and because neurons have these long axons, it's actually some centimeters away from the substantia nigra. And the aim here is really to get an endogenous release of dopamine in the brain. This is to solve one of the many issues that there are with Parkinson's disease. And when we look at managing motor symptoms in Parkinson's disease, those of you in the room who are very familiar with the disease may recognize this symptomatic picture, which is this problem that when you take either L-DOPA or other dopamine modifying drugs, you can be in a period of on where you actually feel very well. You don't have maybe so many symptoms and you don't have many side effects either. But as you progress through the disease, this therapeutic window, as we call it, becomes more and more narrow, and you need maybe more drug to get rid of the symptoms, as well as you easier get side effects of the drugs. And this is because now you don't have much dopamine neurons left that are here to buffer the normal levels of dopamine in the brain. So what we're trying to achieve by transplanting new dopamine neurons is to avoid this fluctuation in dopamine that you get through medication, and to get a more stable endogenous release of dopamine that regulates itself, so you don't get too much or too little dopamine, which is often the problem in these medication regimes. So just to take a step back, because I'm talking to you about cell therapies and stem cells. And when you hear about stem cells in the media, in fact, stem cells can be many different things and can be used for many different diseases. There is no such thing as one universal stem cell therapy. It's many different types of therapies. And when we talk about therapies for Parkinson's disease, there have also been taken different approaches. So I just want to specify to you what we're talking about today. There are trials and also private clinics, which are unfortunately not very well regulated, that offer these stem cell therapies where they're simply just injected into the blood. Now, this is very easy to do, and that's why many people like to do it, because it's not a big hassle to inject some cells in the blood. And the cells that are often used are called mesenchymal stem cells. And these are actually kind of like a stromal cell, a cell that resides in your under your skin, and can be in your bone marrow too. And the reason people inject these is because there is this hypothesis that they have this immune dampening effect, and that this somehow can stop the inflammation in the brain that may be part of the cause of Parkinson's disease. I have to say, though, that there is no evidence, neither preclinically or clinically, that these types of therapies can work. So please do be somewhat skeptical and vigilant if you see a private clinic that is offering to treat your Parkinson's disease by simply injecting stem cells into the blood. We have no reason to believe that this can work, unfortunately. Then there are more complicated administrations where you go into the brain. And of course, the reason we want to go into the brain is because this is where the pathology resides. And here again, you can find two different types of therapies. There are some trials that transplant what we would call more generic neural stem cells. And the idea here is not to make new dopamine neurons, but rather that they secrete some positive factors that can somehow nurture the dopamine neurons and prevent them from dying. There is, for instance, a trial in Australia ongoing with this type of therapy, but again, we do not see very strong preclinical or clinical responses, probably because it is difficult through only this type of therapy to rescue the dopamine neurons. What I'm talking to you about today is what we call dopamine cell replacement. And this means that we are specifically replacing the dopamine neurons and the functionality of the dopamine neurons. So what we're putting in cannot just be any stem cell. It has to be specifically a cell that produces dopamine and can integrate into the dopaminergic circuitry, just like an endogenous dopamine neuron would. And actually, this journey of replacing dopamine neurons started many years ago, all the way back in 1987, where some of the first patients were transplanted in Lund in Sweden. This was something that was really spearheaded and pioneered by Anders Björklund, who is here in the room today. And what Anders and his colleagues did back then, and which was really innovative, was to find a source of dopamine neurons to transplant into the brains of patients. At that time, we didn't have stem cell technology, right? That was not available. But what was available was dopamine neurons, like early newborn dopamine neurons that you could extract from fetal brain tissue coming from legally aborted fetuses. So this tissue was anyway going to be discarded. And instead, Anders and his team got permission to subdissect this tiny part of the brain that they knew would give rise to dopamine neurons. And they then put it into a cell suspension and transplanted it into the brains of Parkinson's disease patients. They transplanted in total 8 patients during this period. Here is one such patient that was transplanted in Lund. This is an MPTP patient. Many of you may be aware that MPTP is not natural Parkinson's disease, but is a very severe form of Parkinsonism, which is caused by intoxication of MPTP, a very potent dopamine neuron toxin. It basically kills all your dopamine neurons very quickly. She's one of a group of patients from the Bay Area in the US that were exposed to this toxin because they had been taking synthetic heroin that was contaminated with this particular chemical. We know now, because of these poor individuals, that NPTP is an extremely potent toxin to dopamine neurons. And in fact, today we use it as an animal model. We give it to mice and to kill off all their dopamine neurons. But unfortunately, in this case, it was humans that were the first guinea pigs. So this young woman, she was transported to Lund University Hospital in Sweden and was transplanted with fetal tissue. You can see her here before transplantation without any dopaminergic medication. And she also responded very poorly to dopaminergic medication. She has a very hard time even getting up, standing on her own, holding her balance and walking. Really even simple day-to-day tasks were very difficult for her. This is now the same individual 5 years after transplantation, again, off any dopaminergic medication. So what you're seeing here is really an effect of the transplant, which is helping her regain much of her mobility again. So cases like this clearly showed us that this approach can work. If you do it right, if you have the right cells, if you transplant enough cells, if the cells survive in the brain, they can, in cases like these, elicit a very prominent clinical response in patients. So actually, they went on later in Cambridge and Lund and proceeded to try to repeat this some uh, 20, 30 years, well, actually almost 40 years later, um, to see if they could repeat this in a more controlled study, because the problem was that even though you saw patients like the individual I just showed you, there was also a lot of variability, and there were other individuals that didn't respond as well, probably because of also problems in standardizing the tissue implantation and the tissue quality. So they then initiated a trial called TransEuro, which was led by Roger Barker, who's also here in the room today. And here transplanted an additional 11 individuals in Lund and Cambridge during the period from 2015 to 2023. And what was seen in this trial was unfortunately also a bit variable. There were examples such as this one. This is a participant number 54 who had limited signal on the fluorodopa PET. And if you look overall at the UPDRS scores, again, there was a lot of variability between individuals, some that didn't respond much to the transplantation. But then again, you saw these individuals within the trial that had quite a remarkable response. This is participant number 60, pre-transplantation, 18 months post-transplantation. You see a very clear, very significant increase in fluorodopa PET in this individual, which is something you would never see during a normal disease trajectory. And simultaneously, this individual was also the individual that responded best to the treatment, and actually over the course of 4 years had a decrease in UPDRS score, part 3 off, of around 20 points. So that's very significant. So again, this trial showed us that this transplantation therapy can really work, although it is variable. Overall, what have we learned from these fetal transplantation cases? Well, we have learned that the graft can actually, the new dopamine neurons you put into the brains of the Parkinson's disease individuals can survive for many years. This is an example of a 24-year-old graft of dopamine neurons in the brain of a Parkinson's disease individual, which looks completely healthy. Actually, the dopamine neurons look exactly like we would want them to look in a healthy brain. So we've learned that these neurons can really survive for many years after transplantation, even after we take away the immunosuppression. This is kind of like an organ donation. You receive tissue from a donor, so it's not matched to yourself, but regardless of this, it can survive for many years in your brain. We've also seen in individual patients that although the effect is slow, it can take up to some 3 years before you gain maximal effect. It can also give you substantial long-term clinical benefit when it is successful. But a main problem is that this fetal cell transplantation is variable. It has some ethical, logistical hurdles, and it's really impossible to scale up to many patients. And this leads us to why I am here today, because in the meantime, all these years of developing this transplantation therapy, we got an alternative. The stem cell technology was developed alongside this path of fetal tissue transplantation. And of course, the field now started to wonder, well, okay, could we get rid of this variable source of dopamine neurons, which is difficult to standardize, and instead use some stem cells we can culture in the lab and use them to manufacture dopamine neurons in the lab that we can then transplant into brains of patients. And of course, this would allow us to gain unlimited access to large quantities of cells, much better possibility of controlling the quality, the standard, the yield of the neurons, and even making manufacturing cells for thousands of patients at the same time. Okay, so how do we get access to a magical stem cell like this? Let's look at what we have available in our body. In fact, we do have stem cells available in our body. We do have stem cells, for instance, in our skin, in our blood, muscle, intestines. These are very active every single day. But the problem is these stem cells are already predefined to a certain task. So your stem cells in your skin will make skin and nothing else. Stem cells in your blood will make blood and nothing else. Muscle will make muscle, and your intestinal stem cells will replenish your intestinal epithelium. And they're very good at this, but they cannot suddenly make something else. They cannot start to make heart cells or brain cells. So, what do we do in a case of a tissue, such as the brain, which does not have stem cells and which cannot regenerate itself, which cannot produce new neurons? So, here we need to look for a different source of stem cells. And this is important to keep in mind when we talk about stem cells. There's no one stem cell for everything, they have different potentials and different diseases they can be used for. So, for the brain, we cannot use a stem cell we have in our body. We actually have to go rewind time, go back in development, and imagine that we're turning back time all the way back to the very earliest stages of human development, right after the time when the egg has been fertilized by the sperm cell. At this time point, this is around 4 to 6 days after egg fertilization, we have a tiny structure called the blastocyst. And you can get access to these blastocysts from in vitro fertilization clinics, because in these clinics, you will have couples go there. They will donate egg and sperm. You will have in vitro fertilization that will be used to obtain pregnancies. And now what happens at these IVF clinics is there will be lots of, in many cases, lots of surplus embryos. They freeze down everything they get. All fertilized eggs will be frozen down, kept in the freezer so they can implant them if that should be needed. But when the couple has finished their IVF treatment, either they have the children they desired or they don't want to continue the treatment, then you would typically discard all the fertilized eggs, the embryos that are in the freezer. At this point, the couple can also choose to donate an embryo for research. So instead of implanting the embryo into the uterus for pregnancy, you can take up the embryo from the freezer and you can culture it a few more days in the dish, and then you get to this blastocyst stage I mentioned to you before. And in the middle of this blastocyst stage, you have what we call embryonic stem cells. They're located here. This is a picture of a blastocyst with embryonic stem cells in the middle. And this is just to show you how small this structure is at this time point in development. Really tiny, tiny. You can't even see it with your naked eye. So you can take these blastocysts, put them in the lab, and then you can isolate these embryonic stem cells that are present in the middle. And when you do that, in 1998, it was discovered that you could get these cells to divide indefinitely. So they would just keep dividing, making copies of themselves. And this is now what we call embryonic stem cells. Here you see them in the lab, cultured on a petri dish. And they look actually very boring. This is what you're seeing here is a colony of probably some 1,000 embryonic stem cells, or tiny, tiny cells located very close to each other in the dish. We also call these cells pluripotent stem cells. This is because they have potency to become many different types of cells. So what is so magical about these embryonic stem cells is that these are the only cells that we have access to that can give rise to all tissues of our body, because these cells, present so early in development, are programmed to be able to give rise to an embryo, right? They have the potential to give rise to every single tissue in the human body. And this is what makes them so amazing. So we can put them in the dish in the lab and they will divide many, many times, and we can freeze them down. And we can then share them with our researcher colleagues all over the world. So you actually only need one fertilized egg to get enough stem cells to treat every single patient in the entire world. And that is literally speaking. They will divide indefinitely. So as long as you just have one embryo, you can freeze down as many cells as you want. And then you can start making the tissues that you're interested in, right? Because these cells have the potential to form every single tissue of our body. including the nerve cells that we can't normally make with any other type of stem cell. So this is why these embryonic or pluripotent stem cells are so very special. Today, we can make pluripotent stem cells not just from the fertilized egg, but also through an alternative route, which we now call induced pluripotency. This was a technology that was discovered by a very clever man, Shinya Yamanaka, and he actually won the Nobel Prize in 2012 for this technology because it was such a magnificent scientific breakthrough. And actually, his Nobel Prize was very unusual because he invented this technology in 2006, and already in 2012, only 6 years later, he received the Nobel Prize. That's very unusual. You probably have noticed when you see the prize delegation, their recipients are often very old. I mean, can be, above 90. And that is because the Nobel Prize is given not just for some interesting scientific technology, it's given for impact, right? So you get the Nobel Prize only when your invention has had an impact on society, has really been broadened out. People are using your technology because it's so great. And because this reprogramming technology was so easy to use and so powerful, because what happens now is now you can make a pluripotent stem cell, not just from an embryo, but from any cell. You can take a cell from any individual in this room and make a stem cell out of it. So anyone can actually make their own stem cells. So this is the reason he won the Nobel Prize only 6 years after he invented this technology. And actually, the Japanese trial, the team we also have present here, are using this technology for their clinical trial. Okay, so when we have these pluripotent stem cells, whether they were derived from embryo or whether they were derived through induced pluripotency, it doesn't really matter. They look exactly the same. And when you have these cells, you can now gain access to tissues that you normally don't have access to. These are examples of tissues generated in the lab from pluripotent stem cells. You can make insulin-producing islets, a three-dimensional intestinal tissue, three-dimensional kidney tissue, even a human eye cup that looks exactly like a human fetal eye cup, and a beating fetal human heart. All of these developed from stem cells. It's quite extraordinary. And this was in the beginning really a research tool that was interesting, but it was very difficult to control. It took a while before scientists became good at producing these tissues. But now we're at an interesting time because now we're actually seeing these types of stem cells being used in patients for various different diseases, including type 1 diabetes, heart failure, but also Parkinson's disease. These stem cells are being used to produce exactly the tissues that are needed for that particular disease and then transplanted into patients. And what we're seeing here is a map of all the clinical trials that are ongoing in the world for all these diseases using pluripotent stem cells. Okay, but we're here to talk about Parkinson's disease. So what is the status on Parkinson's disease? So if we look at all the pluripotent stem cell derived trials in the CNS, so in the field of brain diseases, we can see that the majority of the trials that are ongoing for brain diseases are indeed for Parkinson's disease. This is regarded as one of the primary stem cell amenable diseases because we know which neurons are lost. We now know how we can make them in the dish and transplant them to the right place in the brain. Also, it's not that many neurons, actually. It's only around 200,000 to 300,000 dopamine neurons on each side of the brain. And when we talk about cell numbers, that's actually a very small cell number. So it makes it feasible to go in and replace that number of cells. Okay, so now comes the scientific challenge, which is how do you make the dopamine neurons in the dish, given that you're now working with stem cells that could make everything, right? They can make heart cells, or they can make skin cells, or eye cells, or liver cells. And how do you control them so they don't make all that, but make only dopamine neurons? And this is not easy because one thing is making a neuron, but our brains are very complex. They contain more than 1,000 different subtypes of neurons, right? And to treat Parkinson's disease, we're not interested in all the 999 other subtypes. We're interested only in the dopamine neurons. So this is difficult. And really, what you do as a stem cell scientist is it's kind of a little bit about like being a chef in the kitchen. You know, you test a little bit, add a little bit of this, and a little bit of that, and then you see if you get the right thing. Is it the right dish you're getting out in the end? No, it didn't really work out. You got to go back, change the recipe, add different stuff and different ratios and so on, and then you do that for years. Actually, you can keep doing that for years and years, testing some factors. You add them to your cells because the cells now respond to these factors as if they thought they were in the embryo, and then they produce the tissue that you have instructed them to make based on the factors you expose them to. This is quite hard work. In our case, we spent some around 5 years, and not just us, many groups around the world have tried to find factors that can produce these cells. And what we're looking for here are cells that are generated right here in the midbrain of the embryo because we know this is where the dopamine neurons are born. The recipe in our case involves 8 factors in 16 days. That can vary a bit between groups, but overall, this is a kind of the type of protocols that we're generating. Now you can take these cells and you can mature them in the dish, and then you get what looks like a real dopamine neuron. It has the morphology of a dopamine neuron. It expresses all the markers you would expect from a dopamine neuron. But of course, what you're really interested in is if you can take these, we take them at the progenitor stage, which means before they really become neurons, because you can imagine a neuron has a cell body and has these long axons that project from it. And if you try to take it off the dish at that point, you're breaking all their axons and dendrites, and that actually can kill the cell. So you want to take them when they're kind of a teenager dopamine neuron. They're not fully grown yet, but they have decided they will become a dopamine neuron. And this is what we call the progenitor stage. And then you inject this into the brains of animal models to test if they can also survive and produce dopamine in vivo in the brain. What I'm showing you here is an example of a rat brain that we use as a typical model for testing transplantation of the dopamine cells. In this rat, what we do is we actually inject a toxin on one side of the brain, and this now completely wipes out all the dopamine neurons. It's similar to the MPTP that the young woman I showed you before had been exposed to. This is a toxin that will kill all dopamine neurons on one side, and then you have a completely intact dopamine system on the other side, okay? So no dopamine neurons here and normal dopamine neurons over here. Now you can transplant your stem cells into this lesion side to see if you can get dopamine production again. And this is an example of 2 brains here. One brain that we transplanted with dopamine cells that were produced in the lab from human stem cells, and another brain where we left out one of the 8 factors that are included in that recipe I showed you. If you leave out just one of the factors, you still get neurons. There is a graft here, but it doesn't produce dopamine, right? So you get the wrong type of cells. But this is really what we want. We're actually seeing here almost complete restoration of the dopaminergic terminals in this rat striatum. Now, how do we functionally test if these cells are actually producing dopamine? So here we use the same model where we lesion one side and we transplant to the lesion side. And now we give the rat amphetamine. And what happens is that amphetamine is a way to test if you can get dopamine release, because what amphetamine does is it kind of opens up the pores of the dopamine neuron, so they just release all the dopamine they have in them. So if there is a dopamine neuron present, and if it actually is producing dopamine neuron, you'll get all that released at the same time. Now, in this lesioned rat, you only get dopamine release on one side of the brain, right? Because we lesioned the other side of the brain, so there's no dopamine neurons there. And what happens then is we get this rotational asymmetry, because now you have a rat with lots of dopamine on one side, nothing on the other. And this you can see on the motor symmetry of the rat. And the way you see it is actually that the rat starts rotating around itself. If you put it in a bowl, it can only walk in circles, right, in a bowl. And it will now start walking only one way. And that's because of this motor asymmetry. And what we can then see in this motor asymmetry test is when we transplant new dopamine neurons into the lesion side, we get rid of this motor asymmetry, because now the transplant starts to balance out the dopamine and produce dopamine from the human neurons, right? So now you get dopamine on both sides of the brain. So this is not really a model of Parkinson's disease, but it's a model of dopamine function. And that's what we want it to be, right? We want it to show that the transplanted cells actually can produce dopamine. We also transplanted the cells into pigs, and here we used the MPTP, which you saw was the toxin that this young woman was also exposed to. We give the pigs MPTP, and then we transplanted the stem cell derived dopamine neurons into their brains. And then we did PET imaging, because PET imaging is something you can also do in the clinic. Maybe some of you have also tried this. This is actually a way to diagnose Parkinson's disease, because you can see a loss of dopamine terminals by this marker called fluorodopa PET. And we also use PE2I, which is another marker that binds to dopamine neurons. And what you can see in the brains of these pigs is that they start producing dopamine on the transplanted side. So this is exactly what we wanted to see, that these stem cells can produce dopamine. But there is another very important job that dopamine neurons are supposed to do, right? The one thing is they have to release dopamine, and this is what I just showed you they can do. But another very important job of dopamine neurons is they have to reuptake dopamine neurons, because actually dopamine neurons are the only neurons in the brain that can clean up dopamine. They are the only neurons in the brain that can reuptake dopamine if you have too much dopamine in the extracellular space. And this is important. And it's particularly important in the context of treatment of Parkinson's disease, because a typically used drug in Parkinson's disease is L-DOPA. And what L-DOPA does is it actually hijacks non-dopaminergic neurons, and it tricks them to start to produce dopamine. And this is good because, of course, in Parkinson's disease, you have a lack of dopamine in the extracellular space. So you now get these non-dopaminergic neurons to start producing dopamine, and then you get more dopamine in the extracellular space, which is good. But the problem is that you're lacking this normal reuptake that the normal dopamine neurons would do, because they have this role of buffering to always make sure that there is enough dopamine in the extracellular space, but never too much, right? If there's too much, they clean up after themselves, they reuptake it to buffer the amount. But in the context of Parkinson's disease, we don't have this buffering. And that means that you sometimes get too much dopamine in the extracellular space. So we would like to know if the transplanted dopamine neurons can also reuptake dopamine, because this is, of course, important if you want to have normal dopamine homeostasis. And as you probably know, when you have this too much dopamine in the extracellular space, you can get what we call L-DOPA-induced dyskinesias, which is really caused by the excess of dopamine, because there is no proper reuptake. So we asked whether our stem cell-derived grafts could not just release dopamine, but also ameliorate these L-DOPA-induced dyskinesias, which are caused by too much dopamine. And here we collaborated with Emma Lane, who is at Cardiff University. She has a rat model that she first lesions the dopamine system, and then she gives the rat L-DOPA every single day to mimic the clinical scenario of an individual with Parkinson's disease. And what you see in this rat model is that you get these abnormal involuntary movements in the rats after they have been taken L-DOPA for some months. Now we transplanted our dopamine stem cells into these rats, and what you see is that these abnormal involuntary movements actually completely disappear over time. And we interpret that as the graft has the function to actually reuptake dopamine and properly buffer the amount of extracellular dopamine in the brains of these rats. Okay, so with all that functional data, how do you actually get this into patients? Because, of course, we want it to progress and not just be treating rats for the rest of our lives or pigs, but actually getting this to the patient community and hopefully getting it into clinical trial. And then you have to do a lot of tests. So you make your cell product and you have your cell bank, both of the cells before you make them into dopamine neurons, after you make them into dopamine neurons. You do a lot of testing. You test the safety and efficacy in these rat models. You test that the cells are safe and you can store them in the freezer and nothing bad happens to them. And then you write a huge pile of documents, which you send to the regulatory authorities, asking them whether they would agree that you could now take this product and test it in humans. And in our case, we planned a phase 1/2A clinical trial where we asked to dose 4 individuals with what we call dose 1 and 4 other individuals with the double of that dose. So trying to figure out which dose could work better. And on February 2023, after a lot of communication back and forth with the regulatory authorities, we got permission and we dosed the first Parkinson's disease individual in Lund in Sweden with 7 million dopamine progenitors administered in 40 microdeposits across the 2 hemispheres. And this is what it looks like if you are a brave individual, such as Andy right here, who signs up to be part of the STEM PD trial. And thank you for that, Andy. You have to go through all this, right? So many visits to the clinic, MRI scans frequently to make sure that the graft is there, it's safe, there's no bleeding, overgrowth. You have to do PET scanning for DOPA, P2I, and then, of course, you have to do the UPDRS test, and including the painful UPDRS test, where we ask individuals to go off their medication for 24 hours before doing the test. So it's quite an extensive regime, and this is, of course, because we want to test very thoroughly the effects of the treatment. The trial goes for 3 years in total, but we actually have the primary endpoint. So because this is a safety trial, the official part of the trial is actually ended at 12 months. And since we transplanted our last individual, which was Andy, in October last year, we will be reaching the primary endpoint at the end of this year. But there will be 2 additional years where we will be assessing safety because, as I mentioned to you before, the full effect of the graft may not appear before 2 to 3, or between 1 to 3 years after transplantation. If you look at the kind of response we could anticipate from a transplantation like this, it's really a slow response we're looking for here. We know that immunosuppression, we give the patients immunosuppression for the first 12 months, and then we taper it out because we know that the graft can still survive many years in the brain. We've seen this from the fetal transplantations. And then, of course, what you can hope for if this graft starts working is a gradual decrease in dopaminergic medication, hopefully in dyskinesias, if there is dyskinesias, and a gradual decrease in motor symptoms. But really, this can take years to see full effect. So we're not the only ones, as I've also mentioned to you. So just give you a brief overview of other trials that are ongoing. Our trial is still ongoing, and we have not released any data yet. So I'm sorry I cannot show you any data from our trial, but we have other trials where data has been released. These are the trials that are ongoing right now. The Kyoto trial, run by Syra, that has transplanted 7 patients using induced pluripotent stem cells. That was completed last year and has just been published this month. We have the Blue Rock trial, which uses embryonic stem cells, transplanted 12 patients, was completed also last year and just published also this month together with the Kyoto trial. This is our trial. We transplanted 8 patients. We've completed the transplantations, are still in the phase of follow-up, so we will not be completed before end of this year. There is a trial ongoing in China, which we unfortunately don't know a lot about, so I can't really tell you much. They have told us that they will be transplanting 50 patients, but we don't know what the status is of that trial. There is also a trial in South Korea. They have announced that they transplanted 12 of 12 patients, but we have not seen any data yet from this trial. This is a company called S Biomedics. And then there is a Chinese company called Irogene that's running a trial apparently in the US and in China with 46 patients. Again, we have not seen any data coming out from this trial yet. So the two trials that have been published are the Kyoto trials and the Blue Rock trials. This is data from the Blue Rock trial that was released just earlier this month. And they had two doses, low dose and high dose. And you can see the low dose, there is maybe some decrease in UPDRS, but not to a significant degree. And then in the high dose, they have quite a significant decrease going from 49 in UPDRS score, part 3 off, to 25. So that's actually a 50% decrease in UPDRS score in this high dose cohort, which is quite extraordinary. What we're still trying to understand is that the PET signal was not so clear, so that they didn't see a really very clear difference. There was a bit here maybe in the posterior putamen that was increased from baseline to 18 months. But what has been announced now by Blue Rock is that they will actually be going into a phase 3. So this is quite extraordinary. So they're jumping from a phase 1 trial that was just completed and have received permission to go into phase 3. We don't normally see this because normally you have to go through phase 1, phase 2, phase 3. But they've been allowed, based on the safety data from this trial, to go directly to phase 3. And they said that they will start this trial later this year. So this will be very interesting because this will, of course, give us information from many more patients that we had in this initial trial. The Kyoto trial was also, I should almost let Jun present this himself. This is data from the Kyoto trial where they also saw in the majority of patients actually a decrease in UPDRS part 3 off. Over the course of 24 months. You can see this one patient went above, but the majority of the 7 patients were below. And in this trial, they saw a very clear increase in PET signal. So what you're seeing here is images from one individual in the trial, where you see this is pre-transplantation, almost no fluorodopa PET signal left. And what you're seeing here is now a very clear increase in fluorodopa PET signal, which we believe corresponds to the implantation site of the dopamine neurons. So I think this very clearly shows that these cells really can survive and produce dopamine in the brains of human beings, even these dopamine neurons that have been entirely lab grown, right? So entirely produced in the lab, which is quite extraordinary. Okay, so all the trials I showed you here are using what we call allogeneic approach, which means it's kind of, you get a donor tissue. It's not your own cells, right? I told you before that there is this technology to make stem cells from yourself, but that technology was not used in these trials. Even though the Kyoto trial does use iPS cells, it is still a donor cell. So it's one cell used for all the patients in the trial. But we have just last year actually seen approval of 3 additional trials which are using these autologous induced pluripotent stem cells. This means that the stem cells are produced from the patients themselves. So they will take either a skin biopsy or a blood sample, then make those cells from the patient into stem cells and produce dopamine neurons from those stem cells. It's quite a lengthy procedure and it's quite difficult to do, but nonetheless, these 3 groups, 2 at Harvard and 1 is a company, Aspen Neuroscience, are embarking on this difficult journey. We're not sure how many patients have been transplanted. They are aiming for 8, 6, and 9 patients respectively. I do know at least 3 have been transplanted in this trial, but I don't know what the status is as of today. And just to take you through what the difference is between these 2 approaches, when you get cells from a donor, which is not yourself, such as if you got an embryonic stem cell was taken from an IVF clinic, this is from a different individual, right? It doesn't match to your own immune system. Whereas if you make cells from yourself into a stem cell and then make dopamine neurons from that, has the advantage that it doesn't, in theory, require any immunosuppression. And this is, of course, why some groups are attempting this route, because it can have the advantage that you can avoid any immunosuppression during the clinical trials. The drawback of this approach is that you have to manufacture a new product for every single patient, and that is extremely expensive. And it's extremely time consuming. So the question is really whether this, even if it goes through clinical trial, will it ever be viable on the market? Because the price may be so high that only Elon Musk can afford it. And I don't think we want to go there. So most of the trials are using this allogeneic approach where you do get cells from a donor, and it has this disadvantage that you need to give the trial participants immunosuppression. But the big advantage is that you can now produce cells in huge scale, right? You can produce thousands of vials of cells that can be used to treat thousands of patients all in one manufacturing run. And this decreases the cost significantly. And it also, in my view, increases safety for the patient, because then you're sure that the cells you're transplanting into every single patient has been very thoroughly tested. It's very difficult to test a product if you're manufacturing a new product every single time. So, in practicality, this is really the approach that is being used most in the clinic, but there are a few groups that are trying this autologous approach. So, what are we looking into in the coming years, apart from these three autologous trials that were just initiated? Well, we've seen some companies come into play here, looking into the next round of trials. We have Sumitomo Pharma, who have licensed the Kyoto product, so the product that was used in the Japanese Kyoto trial. And they have announced that they are running 2 trials in the US starting this year, with 2 similar products. One of the trials will be using sham surgery, and this is, of course, to get to prepare for a phase 3 that would allow them to get market authorization. And it is so, especially in the US, that to get market authorization, you do need a sham surgery group. So they will never approve a product that has not been tested against the sham surgery group. This is something we discuss intensely in our group of scientists, both from Japan, US, and Europe, whether this is ethically sustainable, right? There's a lot of ethical discussions to be had around sham surgery. And the view in some countries is that it's actually not acceptable at all. Nonetheless, in the US, it is an absolute requirement for you to be able to go to market. So we are seeing trials that are implementing sham surgery. The same applies for Blue Rock Therapeutics. They, as I mentioned to you before, have received permission to go to a phase 3 trial in the US, so jumping from phase 1 to phase 3. They, of course, will also include sham surgery. And it will be, and I think it will be like this for most of the trials, that there will be a crossover design, right? So for the sham surgery group, they will be offered the treatment if they are eligible by the end of the trial. But that still means that you would go through a couple of years with sham treatment. And then we have Novo Nordisk, which is our partner for the STEM-PD product. They have also announced that they are planning to start a phase 2 multi-site trial, but do not yet have a set starting date for this. So I think some interesting developments are happening. We're going to see much larger trials, and these trials will really be focused on generating efficacy data, as opposed to these initial trials, which have been quite small, and of course, with the primary endpoint being safety. And I just want to end off with the last slide, which is about another pluripotent stem cell product, which is not for Parkinson's disease, but just to show you an example of another stem cell product that is going into the brain, not for Parkinson's disease, but for epilepsy in this case, to show you that we have more interesting data coming out from this stem cell technology. This is a trial run by the company Neurona, which is located in California in the US. And what they do in this trial is that they produce another type of neurons from the brain, not the dopamine neurons, but the type of neurons we call inhibitory interneurons. And inhibitory interneurons have this very specific feature that they dampen the activity of other neurons, right? So they send out signals to say, okay, relax, not so much activity anymore. And this is, of course, useful when you're talking about epilepsy, because epilepsy is a disease where you have too much activity and the brain cannot stop firing. And this is why individuals with epilepsy get these cramps. And what they showed in animal models was that they could transplant these inhibitory interneurons into the brains of mice with epilepsy. And they could then completely get rid, or almost completely get rid of the epileptic seizures in these mice. And they have gone into patients now. These are patients that are severely epileptic, have many seizures per month. Several of them have many seizures per day, and they don't respond very well to medication. So, what these patients are offered is really just taking out that part of the brain which is causing the epilepsy. And that's, of course, not very appealing, as you can probably imagine. So, what they did in this trial was offering these patients instead a transplantation of inhibitory interneurons into that site which is causing the epilepsy. And they have released data. This is the latest press release from the company showing that they have achieved a 92% reduction in disabling seizures. I think they've transplanted now close to 20 patients, but only 5 of them are some 7 to 12 months after transplantation. So, what this really shows us is that these neurons can function in the brain. And I think we're seeing a very clear efficacy of these inhibitory neurons in this particular epilepsy trial. We're following this very closely. I think these data are very exciting. And just an end note, where's the future taking us? I've mentioned to you a few times this problem or this issue of immunosuppression. We have to immunosuppress individuals in these trials. There is the option of this autologous making your own stem cells approach, but it's super expensive. And what other groups are looking into, and companies in particular, is developing something what we call, or equivalent of what we call the stealth technology. You've probably heard of these stealth jet planes that kind of fly under the radar. They're not detected by radar technology. And we can use something similar in the stem cell field where you kind of shield the stem cells so they become invisible to the immune system. And this is being approached by several groups in the field where you take away those flags that a cell would normally have to be detected by the immune system. And if we can make that work, it would be quite an extraordinary technology because it means that you can produce with one cell line, you can produce cells for everyone, every single individual, without the need for immunosuppression. And last year, we saw this paper coming out from Sana, which is another group or another company in the field. They showed that they could get this to work in non-human primates and are now entering clinical trial with this approach. So we're very much keeping an eye out for this because I think this would be an amazing future technology. Okay, so what have I told you about today? It was very lengthy, so I'll help you and give a little bit of a summary. Okay, so I talked to you about the fetal tissue trials. They have all been completed. They're not ongoing anymore. In conclusions, we see from these trials that we have some patients with remarkable improvements, but there's still a lot of variability. So we've kind of parked that approach on the side. It was an amazing proof of concept, but we've moved on to the stem cells using the pluripotent stem cells, either from embryonic or induced pluripotent stem cells. 9 clinical trials have been initiated with this approach, 3 with autologous cells, 6 with a donor cell line. 2 of them have been published, as I just showed you before. The Blue Rock trial with 12 patients, the Kyoto trial with 7 patients. We expect more data to come out this year from the Korean trial. We're seeing big pharma companies planning to start phase 2, phase 3 trials this year and next year. So we're, of course, keeping an eye out for this. Will be very exciting. And in the future, what we may be seeing is this hypoimmune stealth technology that can form future therapies without the need for immunosuppression. So, of course, you have to keep in mind projects like these involve a lot of people. I've mentioned to you some of them today. We have Anders Björklund, who really started this whole field, Roger Barker, who is the clinical lead on our STEM PD trial, the whole Lund team, Malin Palmer, who leads the STEM PD project, Gesine and Håkan, who are the neurologists, Hjalmar is the neurosurgeon, and then we've had a very close collaboration with the Novo Nordisk cell therapy team, who has really helped us to bring this therapy into the clinic. I think every academic at some point needs to collaborate with an industry partner in order to really facilitate this to come into patients. Thank you so much for listening. And I forgot to say, most importantly, thank you to the patients, the participants in our trial, of which Andy is one of them. Thank you for being brave.
Do we have any questions? Actually, have we got a microphone, a roving microphone?
No, I can shout.
Yes, fine.
It's great for the recording, actually.
I can repeat the question.
Thank you, Chris.
I'm cheating. Can you hear me? Yeah, because I don't actually have a question. I only have a comment, that was an absolutely extraordinary talk. Thank you so much. You managed to breach that big gap there is between science and the lay. And those of us who don't have a science background, I mean, I understood every word of that. And I'd just like to thank you for, I don't know how you did it. It's extraordinary. And I've never seen a lecture like that before. Thank you.
Thank you. Thank you so much.
How did you manage to avoid primates in Lund? You went straight from rats to humans.
Yes, and that is a very good question. It is actually very different from region to region, whether the regulatory authorities push for primate preclinical data. In Europe, in general, there is a consensus of avoiding primate studies. So they will only ask for it if they think it's explicitly necessary. In a case like this, I mean, primates are relevant, for instance, when you're injecting or you're administering a chemical and you don't know the metabolism rate in the liver, you don't know how it infects the immune system, you don't know how it distributes in the body. There, maybe a primate is more relevant than a rat model or a mouse model. In this case, our argument was, well, we're injecting the cells very locally in the brain. They will only be exactly where we put them. They're not going into the body. They're working locally. And this effect we can mimic just as well in a rodent brain as we can in a primate. And they agreed to that.
Michael, there's a question from Claire behind. No, Claire, you had your hand up first.
Thank you. I've got cheeky 2 quick questions. One of which was, there was heterogeneity in the population who had the different stem cell transplants. Are there any factors that you've identified which are predictive of efficacy in the transplant patients?
We don't have enough data to predict efficacy in the transplant patients because that's too early. We've done a prediction trial or study in the rats. And we actually, in that study, identified certain markers that are expressed in the stem cells that, in our case, predicted whether the graft would be efficacious in this rat model of Parkinson's disease. So those we implemented into our manufacturing. Yeah.
And then my second quick question, if that's okay, is about the autologous stem cells. A person who needs to be having stem cell transplant for Parkinson's inherently has defunct neurons. So I'm just curious about the prospect of having then using your own stem cells, which may also be faulty.
Yes. And I think that's a very good point. And that's maybe also another reason I'm not necessarily a big proponent of this approach. You can argue majority of PD is not genetic, or at least we cannot find a genetic cause. So maybe it's simply aging, environmental factors. And when you then put in new neurons, they can still survive for some 30 years and maybe they will be fine. But we don't know, right? And some individuals may actually have a genetic cause that we just haven't been able to find. And in that case, I don't think you want to use their own stem cells. No.
Agnete, I know you as a curious scientific mind and an authority and a leader in this field. The cartoons that you had in your presentation,
Are they all off?
who did those?
Which ones?
The drawings of the embryonic stem cells going backwards through the embryonic.
Those
states.
come from Euro stem cell, which was an old EU
Okay.
consortium, actually. You can find them online, but I find them very useful. Yeah, I didn't draw them myself. Sorry. I don't have that kind of skill.
Yeah.
In a normal situation, dopamine terminals will have things like autoreceptors on them. Do you find the similar phenomena with the grafts, and is that a possible site for potential neuromodulation?
We do find them. We haven't looked very thoroughly. We find them expressed in the dish when we look at the neurons. But I have to be honest, it's a very good question. And actually, we haven't looked very thoroughly at inhibition through the autoreceptors. That's a good point. We should maybe look more at that. We do see that they're expressed, yes.
I think Jane had a question behind John.
So if I understood correctly, we're about 18 months in, is that right, in terms of the results of these things? So
Yeah.
is it too early to tell? Do we need to get 5, 10 years before we really know for sure whether this is going to work?
In our trial, we are, I mean, Andy is 7 months in, but the first individual is around 2 years in. But of course, we want to collect from all individuals before we release any data. What we see published from Kyoto is 2-year data. From the Blue Rock trial is also 2-year data. I mean, I do think some of these individuals could see a continued improvement even past the 2 years. So we're hoping, of course, you will publish a follow-up study with the follow-up data, but that hasn't been released yet.
You mentioned the ethical problem of sham surgery and that America was definitely going to stay with that and that Europe might be going some different way. How likely is that? Or do you think Europe will align with the USA? That's the first part. And the second part, what risks are actually involved in the sham surgery or in any surgery involving the transplant?
Yeah, so we should have the neurosurgeon answer this, but I mean, they're not going into the brain in the sham surgery. They're putting the participants in anesthesia. And of course, there is the general risk of anesthesia, which is not big, but it's there. And it's for quite some hours, right? Can be between 3 to 9 hours, depending on the trial. And then they make a tiny burr hole in the skull and then close it up. So there's nothing going into the brain. Of course, there is still risks of infection, as you always have. They're not huge, but they are there. The other risk that can be with the sham group is whether you have to immunosuppress them or not. Because this is also a discussion. The immunosuppression can actually be sensed by the participants often, especially when we're talking about steroids. They can actually sense whether they are on steroids or not, and the doctor can see it. So we've also discussed, and this will be implemented in some trials, that the sham surgery individuals will have to take steroids as part of the immunosuppression regimen to be blinded, right? So it's very complex, actually. And of course, is it worth it, right? Can you expose sham surgery patients to both immunosuppression and the anesthesia and all this? And is that ethically viable? I know in Japan, the regulatory authorities will not accept this. So I think many companies will have to run separate trials. One for the US with sham surgery group, and maybe another one in Japan, Sweden. I know the Swedish regulatory authorities are not keen on it either.
Thank you, Dr. Kirkeby. That's a very interesting presentation, very well explained. You touched on the stem cell clinics. You said they're not regulated, which is not great for patient safety. But do they publish their data?
No. No.
So how are they allowed by the MHRA and the UK
They're not
regulatory?
allowed. They're not allowed.
Yeah.
It's a huge problem. They pop up right? They
Yeah.
pop up, and then it's not, it for many years has not been a big focus of the European authorities to close them. Once in a while, they'll hit down on some of them, close them, but then a few months later, they pop up under a different name. And it just takes years before the authorities find them and actually go through all that paperwork to get them closed. And then they're already ready to start a new clinic. They are not legal. And in general, it should be advised that if you are at a clinic and they offer you a stem cell therapy, which is not marketed, right, which is not NIH approved or MHRA approved, and you are asked to pay for it, this means that it's an unproven therapy. And then it's most likely illegal.
It's probably not safe, so we need to
No,
protect our PD community.
exactly. It's a huge problem. Yeah.
Okay, thank you.
Brilliant. Thank you. Thank you. Thank you.
There are some questions online. I don't know if we should.
Okay.
Yeah.
Amazing if you could bear to.
There is a question, why do you need a sham? If a pet can show if the effect of a stem cell transplant, why is it necessary with a sham? That's another good point, because the PET scan is very objective, right? This is normally not, it's not affected by placebo. And we would argue the same thing. PET imaging is very important for us because this is a truly objective measure. The problem is that it's not a measure of clinical improvement, right? So to get a therapy approved on the market, you have to show clinical improvement. And ideally, it has to be patient-reported clinical improvement. And a PET is not that. So therefore, you need sham. There's a question about brain first, body first. How might this impact your research? And I just can say we don't have enough individuals in this trial yet to say that. I know the Kyoto group is looking into whether some individuals are responding better than others. And I think as we get more patient data, we will see this. And then there is a question, have you seen correlation between the disease progress and the response to cell replacement therapy? And here in general, the approach is, or the consensus is that probably early to moderate disease stage is the stage that will respond best to cell therapy. So this is what we're trying to target in these trials. Yeah.
Thank you very, very much.
Yes.